Most fish look forward. This one rebuilt its head so it could stare almost straight up. Its eyes are not round. They are long tubes buried deep inside its skull. The bones of its face stretch and shift to hold them in place. Its field of view narrows to a thin vertical window pointed toward the faintest light above. It sacrificed almost everything else to see in one direction. The telescopefish of the genus Gigantura lives in the ocean's twilight zone, a layer where sunlight is nearly gone, but silhouettes still exist. Down there, looking ahead doesn't help much. Looking up can mean the difference between eating and starving. So evolution made a decision. Instead of giving it speed, instead of giving it wide vision, it redesigned its skull. But extreme specialization is never free. Human exploration rarely reaches it intact. Bringing it upward can damage the very anatomy that keeps it alive. It is not absent from the ocean. It is simply beyond routine access. Tonight, we examine the telescopefish and the strange evolutionary pressure that turned vision into architecture. But before we begin, if you enjoy calm journeys like this, I'd truly appreciate a like or a subscribe. And if you'd be so kind, let me know in the comments, where are you winding down from tonight? And what time is it there? I always love seeing how far these late night explorations travel. Now take a slow breath, and together, let's descend into the twilight zone and investigate the skull evolution bent to survive. Long before a body is shaped by pressure or hunger, it is shaped by light. The ocean arranges itself in bands of brightness, each layer defined not by temperature or depth alone, but by how far the sun can reach. From above, the sea appears unified, a single blue surface stretching beyond sight. Beneath that surface, illumination thins in measured stages, and life follows those gradients with quiet precision. At the very top lies the sunlit realm, where light pours freely and colors remain intact. Here, reds burn clearly, greens shimmer, and shadows are soft. Waves scatter brightness downward, and energy flows generously through plankton and drifting plants. This upper layer is animated by visibility. Shapes are obvious, movement is easily tracked. Predators and prey negotiate in a world where contrast is strong and outlines are sharp. Below this brilliance, the character of the water changes. Between roughly 200 and 1,000 meters begins the mesopelagic, often called the twilight zone. It is not dark in the absolute sense. It is dim, steadily thinning, suspended between clarity and obscurity. Sunlight still enters, but it arrives weakened, filtered by distance, and absorbed piece by piece. Light does not vanish all at once. As it descends, it separates. Red wavelengths disappear first, swallowed within the upper few dozen meters. Oranges and yellows fade soon after. Greens thin gradually. Blue persists the longest, penetrating deeper than any other visible color. What remains at several hundred meters is a cold bluish residue, faint and directionless. By 1,000 meters, daylight survives only as a diluted glow from far above, a suggestion rather than a beam. There are no bright shafts cutting downward, no dancing reflections. Illumination becomes a ceiling rather than a surrounding field. The water holds a permanent dusk. Despite its subdued appearance, this layer is immense in scale and consequence. The mesopelagic spans the globe, wrapping every ocean basin in a vast belt of dim water. Within it lives one of the largest collections of animals on Earth. Small fish, gelatinous drifters, crustaceans, and soft-bodied predators occupy this space in astonishing numbers. Biomass estimates suggest that life here rivals or exceeds that of many surface fisheries, yet much of it remains uncounted. Exploration has been limited by depth and fragility. Nets distort delicate bodies. Sudden pressure changes alter forms that evolve to remain compressed. Remote vehicles provide glimpses, brief windows into drifting assemblies that scatter when approached. The result is an ecosystem sensed in fragments rather than surveyed in full. It would be easy to imagine such depths as sparse simply because they are hidden. In reality, the twilight zone is structured by gradients and migrations, by vertical boundaries that shift over a single day. Organisms rise and sink through these layers in coordinated rhythms, forming moving strata within the larger band of dimness. The water is populated not as a uniform fog of life, but as a layered city suspended in blue shadow. This is the environment that frames the telescopefish. Not a void, not an abyss, but a region defined by diminishing light and dense presence. The rules here are written by attenuation and distance. Visibility narrows, energy thins, every adaptation begins with this steady fading. Orientation determines destiny long before motion begins. In the dim strata already surrounding our drifting subject, direction is not equal in value. Gazing forward offers little guidance, since the water ahead holds almost no contrast, only a uniform haze where edges dissolve before they fully form. Shapes blend into the surrounding blue, and distance disguises movement until it is nearly touching. A hunter that relied on a horizontal view would drift through ambiguity, its senses filled with sameness. Above, however, the faint ceiling still carries a diluted trace of radiance. Against that distant glow, anything passing becomes a mark, a moving interruption written in shadow. Fins cross that pale field like ink strokes. Small bodies etch brief silhouettes. Even the slowest swimmer reveals itself when it rises between the observer and the last surviving light. In such a place, vision gains meaning only when it is aimed upward. This simple shift transforms geometry into opportunity. The vertical axis becomes a corridor of information, while the surrounding directions fade into obscurity. Creatures adapted to this layer often angle their eyes toward that dim boundary, aligning perception with the only backdrop that provides contrast. Their world is organized not around horizons, but around height. Above means signal, level means obscurity, below means darkness. Predators drifting higher cast passing stains of shade before their forms can be distinguished. Prey ascending from deeper water trace softer outlines, delicate but detectable. Each silhouette carries timing, speed, and intention within its outline. A widening shape signals approach. A thinning one signals retreat. In this environment, survival is written in profiles rather than colors, in motion rather than detail. Many inhabitants have accepted this rule and adjusted accordingly. Their pupils tilt, their retinas bias sensitivity toward faint downwelling light. Their posture steadies to keep that narrow window in view. They become watchers of the ceiling, patient interpreters of dim signals crossing a distant screen. Life unfolds above them like quiet theater performed behind frosted glass. Yet one lineage follows this principle with unusual devotion. Its entire visual world contracts and aligns toward that upper glow, as if all other directions were gently folded away. Instead of scanning widely, it commits. Instead of balancing multiple fields, it chooses a single axis and perfects it. The result is not merely an animal that looks upward, but one whose perception is architected around that act. Through such specialization, the surrounding volume rearranges itself. Space is no longer a sphere filled with possibilities. It becomes a column rising overhead, a vertical stage where every meaningful event appears. The hunter waits beneath this column, suspended with restrained motion, reading the passing silhouettes that drift across its sightline. Each shadow is a message, each movement is a clue. What seems sparse to an outside observer resolves into layered activity when viewed from this angle. The water above is rarely empty. Minute travelers cross it, Gelatinous forms pulse through it. Small shoals slide along its heights. All announce themselves the same way, as dark signatures against a paling veil. To watch upward here is to witness a constant procession. In this realm, orientation is not a habit, but a strategy. Vision aimed skyward becomes a net cast into light itself, gathering information before bodies ever meet. The creature that masters this perspective does not chase the world. It lets the world reveal itself in silhouette. Within that quiet discipline lies its advantage, a life tuned to a single direction, sustained by the faint script of passing forms, reading the brightness above as carefully as others read currents or scent. Nothing else matters there now. Inside the skull, the transformation is architectural. Bone does not merely house the eye, it reshapes itself around it, accommodating a structure that stretches backward like a pair of slender cylinders. These are not rounded orbs resting shallow beneath the skin. They are elongated tubes, each one extending deep into the head, aligned with precise intention. The front opening of each tube admits light through a relatively small aperture. Beyond that entrance, the passage narrows and lengthens, creating a chamber that guides photons inward with minimal scatter. The lens is positioned far from the surface, set well within the cylinder rather than near its rim. This distance is not arbitrary. By increasing the space between lens and retina, the eye effectively lengthens its focal path, refining sensitivity to faint illumination arriving from a restricted direction. At the far end of the tube lies the retina, stretched across the inner wall like a delicate lining. Here, photoreceptor cells wait within layered tissue tuned for dim environments. The arrangement favors rods over cones, emphasizing detection over detail. Each captured photon matters. Each signal is amplified through neural circuits adapted to extract contrast from scarcity. The geometry creates a visual field that is remarkably narrow. Instead of surveying a broad panorama, each eye samples a tight cone of space. Peripheral awareness diminishes, edges of the world slipping beyond perception. What remains is an intensified corridor of sight, concentrated along a single axis. In that corridor, even minimal light can be gathered, focused, and interpreted with unusual precision. Such design carries cost alongside benefit. Reduced side vision limits early detection of movement outside the chosen direction. The animal cannot rely on sweeping glances to monitor its surroundings. Its awareness must be coordinated with posture and subtle body adjustments, keeping the tubes aligned where information is richest. Vision becomes deliberate, almost mechanical in its steadiness. The elongated form also stabilizes incoming light. By channeling illumination through a tubular path, stray rays are minimized, enhancing contrast against the dim background. The internal surfaces are adapted to reduce reflection, preventing loss of precious photons. The lens curvature and retinal placement together create an optical system comparable in principle to a compact telescope, scaled to living tissue. This configuration did not arise in isolation. Other deep-dwelling species, such as certain barrel-eye fish, display related adaptations. In them, eyes also elongate and orient upward, sometimes protected by transparent cranial shields. Convergence suggests that similar environmental pressures sculpt similar solutions. When illumination becomes directional and scarce, optical systems tend to narrow and deepen, favoring sensitivity over breadth. Within the telescopefish, the integration is complete. The skull arches to cradle the tubes. Muscles anchor them with subtle control, allowing minute adjustments rather than wide sweeps. Neural pathways enlarge in proportion, dedicating processing capacity to the signals delivered from that confined window. Anatomy, physiology, and behavior align around the same principle. Nothing in this arrangement appears decorative or accidental. Each structural modification reflects incremental selection, generation after generation favoring individuals that could collect slightly more light from slightly greater distance above. Over time, small elongations accumulated, lenses shifted deeper, retinal surfaces adapted to maximize capture. What began as a modest tilt evolved into a full commitment of form. The result is an eye that has relinquished generality in exchange for acuity within a single direction. It embodies compromise shaped by environment, a living instrument refined for a narrow beam of brightness. In the quiet dimness of its habitat, that refinement is sufficient. Structure determines possibility long before motion reveals intention. In this creature, the architecture of the head has been quietly redrawn so that bone itself serves vision. The cranium arches and lengthens, forming a rigid cradle that holds the elongated eyes steady within their sockets. Rather than resting at the front as in most fishes, the orbits rise higher and angle upward, requiring surrounding bones to stretch and thin, like supports repositioned to hold an instrument aimed toward a distant ceiling. Because the eyes occupy so much vertical space, the mouth cannot remain in its usual place. It shifts downward, settling beneath the visual columns, hinged lower on the skull, where leverage favors an upward snap. The jaws extend with a delicate narrowness, and along their edges sit teeth angled slightly back, shaped to secure anything intercepted above. When they close, the motion travels in a swift arc from below to above, a path aligned with the direction of sight. Muscle attachments follow this rearrangement. Fibers anchor along the modified bones, prioritizing precision over force. The strike does not depend on brute strength but on alignment, timing, and minimal resistance from the surrounding water. Even the neck region, subtle and compact, supports this posture, allowing the head to remain tilted without strain while the body drifts in suspension. The body itself reflects the same principle. It narrows and lengthens, reducing drag and conserving energy during stillness. Fins are modest and carefully proportioned, sufficient for balance yet never excessive. Motion becomes economical, almost suspended, as if the animal were a living instrument calibrated for steadiness rather than speed. Each contour avoids turbulence, preserving a quiet stability that keeps the sensory axis fixed. Seen from the front, the arrangement can appear unfamiliar to human perception. Our own faces are organized around forward symmetry, with features aligned along a vertical plane that emphasizes straight-ahead awareness. In contrast, this fish presents a profile dominated by elevation. The highest structures are the eyes, the mouth rests below, and the slope of the skull follows that hierarchy. What seems unusual is simply anatomy answering a different set of spatial priorities. Perception often labels such forms as strange because it measures them against familiar proportions, yet within its own environment, the design is coherent and exact. Bone supports sight, jaws support capture, muscles support stillness. Nothing is misplaced. Each element occupies the position that best serves the upward line along which survival unfolds. The impression of distortion arises not from error in the animal, but from expectation in the observer. Development guides these proportions from early growth. As the young mature, cartilage firms into bone and angles adjust by small degrees, each change preserving alignment between vision and strike. Gradual remodeling ensures that no stage leaves the hunter mismatched to its needs. The head tilts slightly more, the jaws settle slightly lower, and the balance of mass shifts until posture and perception coincide. Even the skin conforms to this structure, lying smooth across the altered frame, revealing the underlying geometry without obstruction. Subtle ridges trace where bone presses close, while softer regions cushion joints and allow minute adjustments. The entire figure appears arranged around a single directive: maintain alignment with the narrow corridor that defines its awareness. Form follows function so faithfully that appearance becomes a diagram of purpose. In still water, it holds this design effortlessly, a structure balanced as carefully as a suspended instrument. Every line, angle, and surface participates in the same quiet intention, demonstrating how profoundly a body can be shaped when sight becomes the axis around which a life is built entirely. Light behaves differently once it enters the sea. Instead of traveling freely as it does through air, it collides with molecules, suspended particles, and drifting matter, each encounter stealing a fraction of its strength. With every meter downward, the loss compounds, not in a steady line but in a quiet curve, a mathematical fading in which brightness diminishes by proportion rather than by step. A descent of only a short distance can reduce what remains to a thin residue. Illumination that seemed present above becomes scarce below, then scarcer still, as if the water were gently closing over it. By the time these depths are reached, the available photons arrive sparsely, scattered and delayed, like faint taps on a distant surface. Vision under such conditions depends less on abundance than on efficiency. The retina adapts by favoring cells shaped for sensitivity. Rod photoreceptors multiply across its surface, slender and numerous, each one capable of responding to minimal stimulation. Cones, which discern hue and fine detail, recede in importance, their talents unnecessary where color seldom survives the descent. In this dim realm, color loses relevance because wavelengths vanish unevenly long before they arrive. Without the full spectrum, surfaces no longer reflect distinctive tones. Everything trends toward muted shades, then toward uniform darkness. What remains visible is contrast alone, the slight difference between a body and the faint field behind it, a difference measured in whispers of light. For an eye designed to function here, gathering light outweighs all other priorities. Sensitivity must increase without introducing distortion. Neural pathways amplify signals while filtering noise, ensuring that random flashes do not masquerade as meaningful forms. The slightest true variation is preserved, carried inward as a precise message about position and motion. Depth, therefore, acts not only as a measure of distance, but as a regulator of perception. A shift downward by a modest span can have visibility, then have it again, until sight approaches its functional limit. At that boundary, detection replaces observation. The task is no longer to see clearly, but to notice that something is there at all. The fish exists within this margin, where illumination hovers just above absence. Its visual tissues operate close to the threshold at which photons can trigger sensation. Chemical reactions inside each rod unfold with extraordinary restraint, tuned so that even a single particle of light can begin a cascade, translating contact into perception. Energy is precious at such depths, and biology avoids waste. Producing pigments for color discrimination would demand resources that yield little return, so the system invests instead in clarity of outline. Edges matter more than patterns. Boundaries matter more than texture. Recognition is achieved through interruption of faint light rather than through decoration. In this physics of near darkness, certainty is assembled from fragments. A passing shape interrupts the dim field for an instant, and that interruption is enough. Signals travel from retina to brain with little delay, forming impressions that guide motion before the moment fades. Perception becomes brief, exact, and sufficient. Here, sight is not a broad window but a disciplined instrument, calibrated to operate where light is scarce and silence is vast. The laws of optics still govern every ray, yet they act softly, shaping a world assembled from the smallest traces. Within that restraint, vision persists, delicate, exact, and perfectly matched to the narrow allowance granted by depth. No excess remains, no brightness is spared, and nothing arrives without consequence. What little light descends is received, measured, and used, until the boundary between presence and darkness becomes a place where perception continues quietly, without sound or spectacle. Night transforms the upper waters long before it reaches these depths. Far above, where dimness softens into darkness, countless small bodies begin to rise. They leave their daytime layers and drift upward in immense numbers, not in panic or haste, but in a steady, collective ascent that repeats with the turning of the planet. The movement is vast beyond ordinary scale, a living tide composed of fish, crustaceans, and gelatinous drifters, all traveling along invisible paths toward distant nourishment. This ascent forms the largest migration on Earth, though it passes without sound or surface trace. Billions participate each cycle. They climb through the water column as evening spreads, guided by internal rhythms older than reefs or continents. Muscles pulse gently, appendages beat in patient cadence, and fragile bodies glide through gradients of pressure and temperature, rising together as if lifted by a slow breath. By the time darkness settles at the surface, the travelers have reached their nightly destinations. There they feed among plankton clouds and microscopic pastures, gathering energy beneath the cover of night. Hours later, as the planet turns and faint brightness begins its return, the procession reverses. The same multitudes descend again, slipping downward through the layers they crossed before, returning toward concealment in deeper strata. From below, this cycle writes motion across the water above. Shapes pass in loose constellations. Lines drift, scatter, and gather again. The entire overhead expanse becomes a shifting field of activity, never still for long. Even when individual swimmers pause, others replace them, so that movement itself seems continuous, like wind through distant grass. The telescopefish remains poised within its chosen depth, attending to this procession. It does not wander widely in search of opportunity. Instead, it observes the passing traffic, reading patterns within the slow parade. Each small traveler that crosses its viewing corridor carries information in its outline and pace. Some signals promise nourishment. Others pass without consequence. Distinction lies in subtlety, and perception sorts them with quiet precision. Feeding aligns with these vertical migrations. When the nightly ascent thickens the waters above, chances increase. When the descent begins, silhouettes thin and intervals lengthen. The hunter adjusts without haste, sustaining its position while the living current shifts overhead. Timing replaces pursuit. Presence replaces chase. The scale of this phenomenon shapes the entire region. Nutrients travel upward inside migrating bodies and return downward within them hours later. Predators follow the motion, prey anticipate it. The water column becomes a layered thoroughfare governed by light cycles and biological clocks. Every participant contributes to a choreography that repeats each day with remarkable consistency. Within that rhythm, the telescopefish occupies a quiet station, neither leading nor resisting the flow. It watches the passage as generations before it have done, sustained by arrivals from above and by departures that thin the stream. Motion never ceases there. The procession continues, measured and immense, carrying life upward and downward in an endless vertical journey. In that gentle traffic lies a kind of constancy. Nothing remains fixed, yet nothing is truly absent. The flow delivers, withdraws, and returns again, a circulation older than memory, steady as rotation itself. The watcher below receives these offerings without leaving its station, sustained by a procession it did not summon and cannot halt. Countless forms pass and vanish, replaced by others following the same ascending and descending routes. Through this continual exchange, the surrounding depths stay supplied with life, and the silent traveler endures within the moving abundance above. Its world is written in these crossings, a living calendar of motion that marks time without sound or light, forever drifting on. Scarcity shapes behavior more firmly than instinct ever could. In these depths, nourishment does not appear on schedule, nor does it linger once present. Sustenance arrives briefly, unpredictably, then passes on, leaving long intervals of absence behind. A body that lives here must be designed not for plenty, but for uncertainty. Within such conditions, hunger is not an emergency. It is a constant background state, a quiet arithmetic running through tissue and blood. Each motion spends fuel. Each pause preserves it. Survival depends on balancing those exchanges with careful restraint. As if life were a flame kept small, so it will last through a long night. The telescopefish carries this restraint in its physiology. Its metabolism proceeds slowly, guided by chemical reactions calibrated for endurance rather than haste. Oxygen is consumed at a measured pace. Muscles release energy in modest increments. Even digestion unfolds gradually, drawing value from each capture with patient efficiency. Nothing in its internal rhythm suggests urgency. Because resources are uncertain, movement must be deliberate. Sudden bursts would squander precious reserves, and there is no guarantee another opportunity would appear to repay that cost. Instead, the body remains poised, fins making minute adjustments that hold position without strain. The posture suggests stillness, yet within that stillness, countless micro-corrections keep balance with the surrounding water. Precision, therefore, replaces speed as the governing strategy. When potential prey enters range, success depends on timing rather than pursuit. The strike must occur once, cleanly, with no wasted effort. Muscles contract along a practiced path, jaws aligning with exactness shaped through generations. If contact is made, energy gained exceeds energy spent. If not, the hunter returns to waiting, its reserves carefully guarded. This economy extends to every system. The heart beats steadily but without excess force. Enzymes operate at rates suited to conservation. Even growth proceeds slowly, as though the body measures each addition against the cost of maintaining it. In such an environment, efficiency is not merely advantageous. It is the boundary between persistence and disappearance. Long intervals may pass without feeding. During those spans, the fish relies on stored lipids and proteins, metabolizing them with quiet discipline. Tissues release what they can spare, sustaining essential organs while postponing any unnecessary expenditure. Activity remains minimal. The animal drifts, watchful yet composed, allowing time itself to assist in preservation. When nourishment finally arrives, the response is controlled rather than frantic. There is no reckless chase, no prolonged struggle. The motion is brief, exact, and sufficient. One successful capture can offset hours of stillness, restoring balance to the internal ledger that governs survival. In this way, patience becomes a form of strength. Such discipline reflects the larger logic of its world. Energy is rare, Appearances are fleeting, and continuation depends on matching behavior to those truths. The telescope fish embodies that match completely. It does not roam in search of abundance. It waits with impossibility, spending almost nothing until the precise instant when spending becomes worthwhile. Through this quiet accounting, life persists where excess would fail. The body remains lean, the motions spare, the senses attentive. Hunger is present, yet it is measured, contained, and understood by the very chemistry that sustains the animal. In that understanding lies endurance, a steady existence balanced between need and restraint, carried forward one careful moment at a time. Nothing is hurried here, nothing squandered. The rhythm of restraint continues without display, written in slow pulses and measured silences. In that restraint, the animal endures, sustained by discipline, shaped through ages, its quiet form remaining where it is, alive within the narrow margin that allows survival each day. Weight accumulates silently in the ocean. It does not fall from above but surrounds from all sides, building with depth in steady increments. For every 10 meters of descent, another atmosphere of pressure settles in, an added layer of force pressing inward. By the time 1,000 meters is reached, the surrounding water exerts more than 100 atmospheres, a compression that would crumple unreinforced metal and shatter rigid containers not built for such loads. Here, pressure is not an occasional stress, but a constant condition. It pushes against skin, bone, and organ without pause. Any structure containing gas would be squeezed relentlessly, shrinking in volume as depth increases. For creatures adapted to these regions, avoiding such vulnerabilities is essential. The telescope fish carries no swim bladder filled with air to regulate buoyancy in the manner of many shallow species. Instead, buoyancy is achieved through subtler means, through tissues whose density nearly matches that of the surrounding seawater. Lipids and specialized proteins contribute to neutral suspension without creating compressible spaces. The body becomes a structure of fluids and soft matrices, yielding slightly under force rather than resisting it. Flexibility replaces rigidity as a guiding principle. Bones are thinner and less mineralized than those of many surface dwellers, allowing slight deformation without fracture. Connective tissues possess elasticity that absorbs pressure rather than opposing it. Organs are compact and arranged to minimize empty cavities, leaving little internal void for compression to exploit. Even the eyes, elongated and delicate in form, are supported by fluid environments that equalize external force. This balance works only within the depth range for which it evolved. If lifted rapidly toward the surface, the surrounding pressure drops far faster than the body can adjust. Dissolved gases expand, fluids shift. Tissues accustomed to compression experience stress in reverse. Structures that function flawlessly at depth may distort or rupture when freed from the weight they depend upon. For this reason, retrieving such animals intact is challenging. Traditional nets raise specimens through hundreds of meters in minutes, exposing them to gradients their physiology cannot accommodate. By the time they reach air, their forms often bear the marks of ascent: expanded tissues, displaced organs, altered contours that obscure the original design. Modern exploration attempts to counter this difficulty. Pressurized chambers and specialized submersibles aim to preserve ambient conditions during collection, yet even these methods face limitations. Maintaining stable pressure from capture to laboratory requires precise engineering, and small errors can have significant effects on fragile anatomy. Within its native layer, however, the telescope fish experiences none of this disruption. The immense force surrounding it is simply the medium of its existence. Every cell is calibrated to operate under compression, every biochemical reaction proceeding in equilibrium with the weight outside. Proteins fold in patterns stable at high pressure. Membranes maintain fluidity through subtle adjustments in lipid composition, ensuring that cellular processes continue smoothly despite the load. Thus, the very factor that would crush surface life becomes neutral here, integrated into the design of muscle, nerve, and bone. Pressure shapes boundaries, but does not hinder movement. It presses evenly from all directions, eliminating collapse by denying imbalance. In this sustained compression, life persists without spectacle. The water bears its mass, the body yields just enough, and equilibrium holds. The environment that would defeat steel supports a creature built not to resist force, but to exist within it completely. Scale is easy to misjudge in the deep. Distance, dimness, and unfamiliar proportions distort expectation, leading many to imagine its inhabitants as small and fragile. Yet the telescopefish can reach lengths exceeding half a meter, its body extending far beyond what its delicate head alone might suggest. It is not a miniature curiosity, drifting unnoticed. It is a substantial presence, elongated and deliberate, occupying its lair with quiet authority. The impression of smallness often arises from the scarcity of direct encounters. Few have observed this fish alive in its natural setting. Fewer still have recorded it moving freely, undisturbed by ascent or capture. The majority of specimens known to science have been retrieved through nets drawn upward from great depth, their forms altered by the journey before they ever meet human eyes. During rapid ascent, pressure decreases and tissues expand beyond their accustomed equilibrium. Eyes that function perfectly below may protrude unnaturally at the surface. Internal organs can shift. The body, built for compression, loses the balance that once held it in precise alignment. What arrives on deck often bears little resemblance to the living posture it maintained in its native lair. Photographs taken under such conditions can mislead, emphasizing distortion rather than design. Limbs seem slack, proportions exaggerated, textures softened by decompression. These images circulate widely, shaping perception of a creature that few have seen intact. The result is an impression of fragility or strangeness detached from the context that explains it. Occasional glimpses from submersibles offer a clearer view. When encountered at depth, the telescopefish appears composed and streamlined, its length apparent as it hovers with measured stillness. The head, though prominent, integrates smoothly with the tapering body behind it. Fins balance the form without excess. The overall silhouette reveals an animal neither diminutive nor grotesque, but proportioned according to the logic of its environment. Still, such footage remains rare. Deep expeditions are costly and limited in duration. Vast expanses of ocean remain unexplored, and the mesopelagic layer spans the globe. A half-meter fish drifting within that immensity can easily pass beyond the narrow beams of artificial light carried by research vehicles. Encounters depend on timing, location, and chance alignment. Because direct observation is scarce, knowledge accumulates gradually through fragments. Measurements taken from preserved specimens provide length and anatomical detail. Tissue analysis reveals composition and structure. Comparisons with related species fill certain gaps, yet behavior, population size, and daily rhythms are inferred more often than witnessed. The living giant remains partially concealed within its own vast domain. Its size carries ecological significance. A body of that length requires sustenance sufficient to maintain muscle, organ, and bone. It occupies a place in the food web proportionate to its dimensions, neither at the smallest margins nor among the largest predators. Its presence reflects a balance within the twilight community, where medium-sized hunters contribute to the layered hierarchy of consumption and survival. To consider the telescopefish as a substantial animal rather than a tiny anomaly shifts perspective. It is not a minor experiment of evolution tucked into darkness. It is a mature design extended across measurable length, capable of enduring the pressures and scarcities already described. The rarity of intact observation does not diminish its scale. It simply reminds us how little of that realm has been seen directly, and how much of its true form continues to reside beyond routine view. Steady and full-sized within the dim expanse it calls home, discovery did not arrive as a single moment, but as a gradual unveiling shaped by patience and machinery. Long before cameras descended into these depths, knowledge of such creatures began with nets lowered into darkness, drawn slowly through layers few instruments had touched. These deep-sea trawls traveled unseen paths, their mesh mouths open to whatever drifted within reach, then rose again bearing fragments of a hidden community. Among those early collections were forms unlike any known from shallower waters. Some appeared elongated, some translucent, others bearing proportions that seemed improbable under ordinary conditions. The telescopefish entered human awareness through such encounters, its presence revealed not by observation in motion, but by retrieval from pressure into air. Scientists studying these specimens relied on careful measurement and comparison, piecing together anatomy from bodies already altered by ascent. The act of collecting from depth imposes unavoidable strain. Nets must travel long vertical distances, and during that journey, conditions change continuously. Temperature shifts, pressure falls, and delicate tissues respond to each transition. By the time a specimen reaches the surface, its structure may have expanded or slackened, creating contours that differ from its living state. Researchers must interpret these altered shapes with caution, distinguishing natural design from distortion caused by retrieval. Handling such fragile animals requires restraint and specialized methods. Even gentle contact can damage tissues adapted for environments where external forces remain constant. Preservation often begins immediately, stabilizing form before further change occurs. Fluids are sampled, bones examined, and microscopic structures studied under magnification. Through these procedures, details emerge that reveal how the organism functioned before it ever entered the net. Technological advances introduced new approaches. Remote-operated vehicles, guided from ships above, descend carrying lights and cameras sealed within pressure-resistant housings. These machines move deliberately, allowing scientists to observe animals in their native surroundings without bringing them upward. Through their lenses, the telescopefish has occasionally been seen suspended in its natural posture, its form intact and its movements unhurried. Such footage is uncommon. The deep ocean covers most of the planet, and expeditions can survey only small regions at a time. Visibility is limited to the reach of artificial illumination, which reveals only a narrow corridor through the vast water. Many animals drift beyond that beam before they are noticed. Encounters depend on alignment between vehicle and organism within an immense volume of space. When images are captured, they sometimes appear unusual to viewers unfamiliar with deep-sea optics. Artificial lights can flatten texture or exaggerate reflective surfaces. Lens curvature may alter proportions near the frame edges. Particles suspended in water scatter illumination, creating halos or specks that obscure fine detail. Scientists account for these effects when analyzing recordings, adjusting interpretation to match physical reality. Over time, each observation adds a fragment to a larger understanding. A preserved specimen contributes measurements. A video sequence reveals posture or motion. A tissue sample discloses biochemical adaptation. Gradually, these pieces assemble into a portrait of a species seldom witnessed directly. Knowledge grows not through constant visibility, but through careful accumulation of rare evidence. Thus, the telescopefish entered scientific awareness through instruments rather than casual sight. Nets first revealed its existence. Cameras later confirmed its living form. Between those glimpses lies a discipline of inference, where researchers reconstruct life from traces gathered at the boundary between depth and air. The result is a quiet partnership between technology and curiosity, allowing study of a creature that remains, for the most part, beyond ordinary reach. Elsewhere in the dim water, other forms hold the same quiet posture, their attention directed toward the faint brightness above. They belong to different families, shaped by separate ancestries, yet their silhouettes reveal a shared intention. Nature has guided them along parallel paths, each lineage arriving at similar solutions through its own slow history. The barrel-eye fish drifts with transparent calm, its head encased in a clear dome through which its eyes can be seen. Those eyes angle upward, enclosed and protected, scanning a narrow region where passing bodies may cross. Though its shape differs from that of the telescopefish, the purpose is closely aligned. Both rely on a focused visual corridor that prioritizes detection from above rather than around. Nearby, hatchetfish maintain a comparable vigilance. Their bodies are thin and reflective, laterally compressed, like blades suspended in water. Large eyes dominate their heads, tilted toward the same distant source of illumination that governs perception throughout this depth. The reflective surfaces along their flanks reduce their visibility from below, while their upward gaze ensures they can register faint movement crossing the brightness overhead. Dragonfish follow a more sinuous design. Their elongated forms carry rows of delicate photophores along the sides, organs capable of producing controlled light. Even with this ability, their eyes remain attentive to the region above, where silhouettes provide advance notice of opportunity. Their teeth extend in slender arcs, adapted for swift closure when a suitable shape enters reach. Though their appearance suggests a different strategy, the underlying orientation is familiar. These animals are not close relatives in the evolutionary sense. Their lineages diverged long before their present forms existed. Yet the environment they share imposes consistent demands, and those demands shape bodies in comparable ways. This process is known as convergent evolution, a pattern in which unrelated species develop similar traits because they face similar challenges. The resemblance does not arise from common ancestry, but from common necessity. Within this depth, the challenge is clear. Vision must function where light is scarce and directional. Prey and predator alike must interpret faint signals quickly. Species that orient their sensory systems toward the most informative region gain advantage, and over generations that advantage becomes embedded in anatomy. Eyes tilt, skulls adjust, muscles refine, and behavior aligns until different creatures exhibit parallel adaptations. Observing these hunters together reveals a quiet unity. Each appears distinct in outline and texture, yet all share the same upward attention, as though listening for a distant sound. The water column above them acts as a stage on which brief forms appear and vanish. Their senses remain tuned to that stage, ready to translate passing shadows into action. This shared strategy does not erase individuality. The barrel eye retains its transparent shield, the hatchetfish its mirrored sides, the dragonfish its luminous organs, and the telescopefish its elongated visual structures. Diversity persists within convergence, allowing each species to occupy a slightly different niche while still responding to the same physical realities. Such repetition of solutions across unrelated animals reflects a broader principle within evolution. When conditions remain stable over immense spans of time, certain designs prove consistently effective. Natural selection favors those designs again and again, shaping new bodies along familiar lines. The result is a community of organisms that appear to echo one another, not through imitation, but through adaptation to identical constraints. In this layered world, the upward hunters drift in separate paths, yet share a common orientation, each one a variation on a theme written by depth and light, their differences resting lightly upon a foundation of the same quiet logic. In much of the deep ocean, light is not merely received, it is made. Countless species generate their own illumination through chemical reactions housed within specialized organs. Blue flashes pulse along flanks, small lanterns shimmer beneath jaws, delicate sparks flicker from drifting bodies. Bioluminescence serves many roles, signaling mates, confusing predators, luring prey. In a realm where sunlight fades, living light becomes common currency. Against this luminous backdrop, the telescopefish remains dark. It carries no rows of photophores along its sides, no dangling lure to entice curiosity, no steady glow to outline its form. Its body reflects only what little illumination arrives from elsewhere. This absence might seem a disadvantage within a community that so often writes in light, yet it reflects a deliberate economy. Producing light requires energy. The chemical reaction underlying bioluminescence depends on substrates such as luciferin interacting with enzymes like luciferase, a process that consumes metabolic resources. Even when highly efficient, each flash or glow draws from reserves that must be replenished through feeding. In a setting where nourishment is uncertain and intervals between meals may stretch long, every expenditure carries weight. The telescopefish avoids this cost entirely. By relying on ambient light rather than generating its own, it conserves the fuel that others spend in signaling or deception. Its visual system is tuned to detect silhouettes against faint brightness, allowing it to harvest information without advertising its presence. Darkness becomes a form of concealment, as well as a strategy of conservation. Active illumination can reveal more than intended. A glowing organ may attract prey, yet it can also betray position to larger predators attuned to the same signals. The deep ocean is not empty of watchers. Many species perceive bioluminescent cues with remarkable sensitivity. To remain unlit is to reduce visibility within that complex network of observers. Passive hunting aligns with the telescopefish's broader pattern of restraint. Rather than drawing attention through light, it waits for movement to cross its field of view. The overhead procession supplies potential meals without the need for active lure. Precision replaces display. Timing replaces invitation. In this exchange, minimal effort yields sufficient return. There is also subtle physics at play. Artificial light, even in small quantities, scatters in water and may create halos that obscure fine contrast. A hunter dependent on reading faint outlines benefits from a clear optical path. By remaining unilluminated, the telescopefish preserves the purity of the dim signals it interprets, avoiding self-generated interference within its own visual corridor. This choice does not imply inferiority to luminous neighbors. It represents an alternative solution to the same environmental constraints, where some species invest in chemical brilliance, others refine sensitivity. Both approaches can succeed, depending on posture, prey type, and energetic balance. In this case, the emphasis falls on perception rather than projection. Over evolutionary time, individuals that conserved energy without sacrificing feeding success would have held advantage. Gradually, traits favoring passive detection strengthened, while those associated with light production never developed. The result is a predator that operates quietly, its effectiveness derived from alignment with existing illumination rather than from the creation of new light. Within the layered community of the twilight zone, this absence of glow is not an omission but a statement of strategy. The telescopefish participates in the dim exchange without contributing to the sparkle that surrounds it. It watches, it waits, and it acts when necessary, sustained by a method that expends little and reveals less. In a world where many creatures shine, this one remains dark by design, its silence in light echoing the broader discipline that defines its form and behavior, conserving strength while others flicker and fade around it. Visibility can be reduced not only by darkness, but by absence. In certain inhabitants of this depth, flesh itself becomes faint, as though matter has softened into glass. Muscles lose heavy pigmentation, scales thin, and internal structures align in ways that allow light to pass through rather than bounce back. These bodies do not reflect strongly, do not cast firm outlines, and so they slip quietly into the surrounding water, present yet difficult to distinguish. Transparency functions as a form of camouflage shaped by physics rather than color. When tissue transmits light instead of scattering it, the boundary between organism and environment weakens. Edges blur. Motion remains the only reliable clue that something is there. Many twilight species rely on this subtle disguise, their forms composed of gelatinous matrices and sparse pigments that minimize visual interruption. In such bodies, even organs may appear suspended, faintly visible yet softened, like shapes seen through misted glass. Others employ a different method. Along their undersides, specialized cells generate gentle luminescence that matches the faint illumination descending from above. This process, known as counterillumination, balances brightness so precisely that the animal's silhouette dissolves when viewed from below. Predators searching upward perceive only uniform glow rather than a solid shape. The organism does not vanish completely, yet it merges with the background closely enough to escape notice. Achieving such balance requires careful regulation. Photogenic tissues adjust intensity in response to changing light levels, brightening or dimming to maintain equivalence with the surrounding field. Neural signals coordinate this output, ensuring that the glow remains steady rather than flickering. The effect is quiet and continuous, more like breathing than shining, a soft radiance tuned to conceal rather than reveal. Blending from below holds particular importance in this vertical world. Many hunters search upward for passing forms, interpreting contrast as evidence of life. Any creature whose underside appears darker than the water above risks detection. By equalizing brightness, counter-illuminating animals erase that contrast, replacing their shadow with imitation light. They become visual echoes of the environment itself. Transparency and controlled luminescence represent two approaches to the same challenge. One removes reflection, the other replaces shadow. Both reduce the chance of being recognized by eyes attuned to subtle difference. Survival depends not only on perceiving others, but on avoiding perception in return. In these depths, invisibility can carry as much value as sight. The The telescopefish moves within a community shaped by such strategies. Around it drift neighbors that scarcely register against the dim background, their outlines dissolving before they fully form. Some appear only when they turn, revealing a brief glimmer of internal structure. Others seem to vanish entirely until they shift position. The water holds them gently, and their concealment remains intact until motion betrays them. These adaptations arise through gradual refinement. Individuals whose tissues scattered less light or whose glow matched ambient levels more closely would have avoided detection more often. Over generations, those traits accumulated, producing bodies increasingly suited to concealment. The result is a population whose members approach visual neutrality, neither bright nor dark, but balanced between. Within this delicate equilibrium, perception becomes a contest of subtleties. Hunters rely on minute deviations, prey rely on minimizing those deviations. Each refinement in detection encourages a refinement in disguise, and so the cycle continues, shaping anatomy through quiet competition. Nothing dramatic announces these changes. They unfold slowly, guided by the steady logic of survival. Thus, the twilight community contains creatures that nearly disappear while fully present. Their existence reminds us that form does not always seek attention. Sometimes it seeks absence, shaping itself until it resembles the medium that surrounds it. In such bodies, concealment is not an accessory, but a fundamental design, as essential as muscle or bone, allowing life to persist within a realm where being seen can mean being taken. Posture becomes perception over time. When a body spends its life aligned in one direction, the world organizes itself along that axis. For the telescopefish, existence unfolds not across a broad horizon, but along a column rising above. Its muscles, bones, and nerves have long since adapted to this orientation, and with them a quiet behavioral pattern has settled in place. The head tilts upward as a matter of course. Fins stabilize the body so that the gaze remains fixed along its chosen corridor. There is little incentive to turn widely or scan in arcs. Instead, attention gathers into a single, steady line, extending from the eyes toward the dim expanse overhead. Movement outside that line holds less relevance. Information arrives primarily from above, and so awareness condenses there. Living beneath larger forms reinforces this vigilance. The upper waters contain not only prey, but also animals of greater size, silhouettes broad enough to dim the faint brightness as they pass. A creature positioned lower in the column must interpret each descending shape carefully. Some outlines promise sustenance, others signal risk. The difference may rest in subtle variations of speed or contour, distinctions that demand continuous focus. This sustained attention shapes behavior in ways that resemble habit. The fish does not dart restlessly, nor does it drift inattentively. Its posture suggests readiness held in reserve, as though every moment might carry significance. Even stillness contains alertness, a quiet calibration of sight and body aligned toward the same narrow window. Such a life compresses experience. Rather than inhabiting a spherical world rich with lateral detail, the telescopefish navigates a vertical narrative. Events appear, cross the visual field, and disappear. Time is measured in passages above, in intervals between crossings. The surrounding water may extend endlessly, yet meaningful occurrences concentrate within that confined region. This orientation may appear limiting when imagined from a human perspective, yet within its environment, it provides clarity. By excluding less informative directions, the fish reduces distraction. Neural processing can prioritize contrast within its chosen corridor, refining interpretation of slight changes. Efficiency of attention becomes an adaptation as vital as bone or muscle. The mental image that arises is one of constant sky watching, though the sky here is water layered with faint illumination. The fish occupies a position analogous to an animal resting beneath open air, eyes lifted toward movement across the firmament. Yet, unlike surface dwellers who occasionally glance upward, this posture is continuous. There is no turning away from it, no alternate vantage that offers comparable value. Over generations, individuals that maintained this unwavering alignment likely fared better than those whose focus wandered. Slight improvements in reaction time or accuracy would accumulate, favoring descendants with heightened vertical awareness. Gradually, the pattern becomes ingrained, a behavioral inheritance as steady as anatomy. Within this disciplined perspective, life proceeds without spectacle. The world narrows and deepens simultaneously, concentrating meaning into a thin shaft of space. The telescopefish endures by inhabiting that shaft completely, neither distracted by what lies beyond, nor unaware of what passes through. Its existence is defined by attention, a steady, upward regard that transforms a dim region into a stage of continual significance. In this way, psychology and physiology converge. The architecture of the skull supports the angle of sight, and the angle of sight shapes the rhythm of living. A narrow visual corridor becomes an entire world, sufficient and encompassing, guiding motion, restraint, and response. Beneath the layered waters, the fish continues its quiet vigil, sustained by the simple discipline of always watching above. Beginnings rarely resemble outcomes in the ocean. The telescopefish does not emerge from its egg, already equipped with elongated eyes and a skull angled toward the dim ceiling of its adult world. Its earliest form drifts closer to the surface, small and translucent, shaped more like countless other larval fishes than like the specialized hunter it will become. At that stage, its proportions are balanced, its head compact, its gaze not yet committed to a single direction. Larval life unfolds in brighter waters where food is more abundant and predators differ in kind. The young fish feeds on microscopic prey, growing through incremental changes guided by genetic instruction and environmental signal. Its eyes are relatively round and positioned in a manner that allows broader scanning. In this early interval, versatility serves better than specialization. The world around it demands general awareness rather than narrow focus. As growth continues, subtle shifts begin. Cartilage strengthens and gradually ossifies. The head elongates in ways that are almost imperceptible from day to day, yet significant over months. Eye sockets angle slightly upward. The spacing between lens and retina increases as the ocular tubes extend. These transformations occur not as abrupt redesigns, but as steady refinements, each stage building upon the last. Habitat depth changes alongside anatomy. Juveniles begin descending gradually from the brighter layers of their birth toward the dimmer zones that will define adulthood. This movement exposes developing tissues to new pressures and light conditions. The reduction in available illumination favors individuals whose visual systems can capture faint contrast more effectively. Growth and environment interact, each reinforcing the direction of change. The reorientation of the eyes reflects this descent. As the fish settles deeper, upward viewing becomes more informative than lateral scanning. Development adjusts accordingly. Muscles adapt to support the evolving posture. Neural pathways reorganize emphasis toward the signals arriving from above. The skull reshapes to cradle the lengthening ocular structures, aligning bone with function in progressive stages. This transformation exemplifies how ontogeny mirrors ecological demand. The larva begins in a realm where generalized perception suffices. The adult inhabits a layer where specialization yields advantage. Between these states lies a corridor of change, guided by both inherited pattern and environmental feedback. Selection does not merely sculpt across generations, it also shapes the timing and sequence of growth within a single life. Throughout this process, energy allocation shifts. Early development prioritizes rapid growth and dispersal. Later stages invest in refinement of sensory architecture and efficient metabolism. The body reallocates resources as priorities change, emphasizing structures most relevant to the depth it now occupies. What was once unnecessary becomes essential. By the time maturity is reached, the fish bears little outward resemblance to its larval self. The elongated eyes dominate the head, the jaw settles beneath them, and the posture reflects commitment to a vertical field. Yet the earlier stages remain embedded in its history, silent reminders that specialization is achieved gradually rather than granted at birth. This developmental journey illustrates a broader principle of life in the ocean. Environments vary with depth and time, and organisms respond not only through evolutionary lineage, but through staged transformation. The telescopefish becomes what its adult world requires through a sequence of measured alterations, each aligned with the pressures encountered along its descent. In tracing this arc, from generalist larva to specialized hunter, one sees how growth itself becomes adaptation. The form is not fixed at emergence, but refined through experience and exposure until anatomy and habitat correspond precisely. Development does not resist the environment. It listens, adjusts, and ultimately arrives at the design that endures within the dim layers below. Currents carry more than water through the ocean. They move elements, compounds, and invisible exchanges that link distant layers into a single system. Among the quiet participants in this circulation are the midwater fishes whose daily routines shift material from one depth to another. Their journeys create pathways through which carbon travels, descending gradually toward the deep. Carbon enters the sea, largely through microscopic plants near the surface, where sunlight allows photosynthesis. These phytoplankton absorb dissolved carbon dioxide and convert it into living tissue. When they are eaten, that carbon passes into the bodies of small animals, and from them into larger ones. Each transfer carries the element farther from its atmospheric origin, embedding it within living matter. Mesopelagic fishes participate in this exchange as mobile links. Many feed in regions above their daytime residence, taking in prey that contain carbon fixed near the surface. After feeding, they return downward, carrying that material within muscle and organ. Digestion redistributes it through their bodies, and metabolism converts portions into energy, releasing the rest as waste products or respired compounds. Those byproducts do not remain suspended indefinitely. Particles sink slowly, settling through layers until they reach deeper waters where decomposition proceeds at a measured pace. Some carbon becomes incorporated into sediments on the seafloor, effectively removed from rapid exchange with the atmosphere. In this way, biological activity assists in transporting carbon from upper layers into long-term storage. Scientists describe this process as part of the ocean's carbon pump, a system driven by both physics and life. Gravity pulls organic fragments downward, microbes break them apart. Animals accelerate the transfer by ingesting, transforming, and releasing matter at depth. Each contribution may be small, yet the collective effect across billions of organisms becomes substantial. The telescopefish belongs to this broader network, even though it neither produces carbon nor consumes it directly from the air. By feeding on other animals and processing what it consumes, it participates in redistribution. Its waste descends, its respiration releases dissolved carbon compounds, and its body stores elements that once drifted near the surface. Through ordinary survival, it becomes part of a planetary cycle. This connection links individual behavior to global consequence. Feeding choices influence how much carbon travels downward. Population sizes determine the scale of transport. Variations in migration patterns can alter where carbon is released. Though no single fish shapes the climate, the combined activity of countless individuals contributes to the regulation of atmospheric balance over long periods. The efficiency of this living pathway depends on stability within the midwater community. If numbers decline or distributions shift, the flow of carbon may change as well. Researchers study these patterns carefully, measuring how much organic material sinks and how quickly it travels. Instruments track particles, analyze chemical signatures, and estimate the role played by animals moving through different depths. Such investigations reveal that life and climate are intertwined through subtle exchanges. Carbon that might otherwise return swiftly to the atmosphere instead descends, guided by biological processes. The ocean thus functions not only as a reservoir, but as an active transporter, moving elements along routes shaped by feeding and metabolism. Within this vast system, the telescopefish drifts almost unnoticed, one participant among many. Its actions are simple: eat, digest, release, yet the consequences extend far beyond its immediate surroundings. Each meal becomes part of a slow descent, each exhalation part of a chemical balance maintained across oceans and years. Seen in this light, the quiet hunter contributes to a planetary rhythm. Its existence supports a chain of transfers that connect surface air to deep water, linking sky, sea, and sediment through living motion. The pathway remains invisible to casual view, yet it continues steadily, sustained by countless bodies carrying carbon downward through the dim layers of the world. Breath exists even where it cannot be seen. Dissolved within seawater, oxygen drifts between molecules, available to any organism equipped to draw it across delicate membranes. Its concentration, however, does not remain constant with depth. In certain midwater layers, the supply thins, not absent but reduced, forming regions where each intake delivers less than it would nearer the surface. These zones of lower oxygen arise through a balance of physics and biology. Surface waters absorb oxygen from the atmosphere and from microscopic photosynthesis. As organic matter sinks and is consumed below, respiration by countless organisms gradually uses that oxygen faster than it can be replaced. Circulation may be slow and renewal limited, leaving pockets where availability declines. Life persists there, yet only species able to function within narrow margins can endure. The telescopefish inhabits such margins with quiet efficiency. Its gills present wide, delicate surfaces through which dissolved gases diffuse into the bloodstream. Thin filaments and dense capillary networks maximize contact between water and blood, allowing extraction of oxygen even when concentrations are modest. Circulation distributes that oxygen carefully, delivering it first to organs whose activity must never cease. Metabolism adjusts to match supply. Chemical reactions proceed at measured rates, conserving oxygen as if it were a rare resource. Muscles contract without extravagance. Digestion unfolds slowly, timed to avoid sudden demand. Even neural activity reflects restraint, transmitting signals with precision rather than excess. In this way, the body aligns its needs with what the environment can provide. Such balance leaves little room for error. Should oxygen levels fall further, tolerance may be tested. Many deep-dwelling species occupy ranges defined partly by this constraint, their distribution tracing invisible boundaries where concentration shifts from sufficient to scarce. These boundaries can change with currents, temperature, and biological activity, altering the habitable space available to organisms adapted for specific conditions. The margin between survival and stress remains slender. A slight decline in dissolved oxygen can influence behavior, slowing movement or reducing feeding. Extended exposure to lower levels may strain physiological systems, forcing adjustments that consume stored energy. Creatures adapted to stability depend on that stability continuing. Environmental shifts, therefore, carry significance. Variations in ocean temperature affect how much oxygen water can hold, while changes in circulation influence how quickly it is replenished. If upper layers warm or currents alter course, deeper regions may receive less renewal. Scientists observing global patterns note that some low oxygen zones have expanded in recent decades, a trend linked to climate dynamics that modify ocean chemistry and flow. For inhabitants of these depths, such trends represent quiet pressure rather than sudden upheaval. There is no visible signal, only a gradual thinning of what they breathe. Adaptations that once matched conditions precisely may approach their limits. Some species might adjust by shifting depth or altering activity. Others may find fewer suitable areas remaining. The telescopefish persists within its established range, sustained by mechanisms refined over generations. Its blood binds oxygen efficiently through specialized hemoglobin molecules that capture and release gas with sensitivity suited to low concentrations. This biochemical tuning complements its restrained metabolism, forming a coordinated system designed for scarcity. Life here is therefore defined by balance. Oxygen is present but never abundant. The animal neither wastes nor hoards it. Each breath drawn across the gills becomes part of a measured exchange, sustaining tissues without surplus. Within that equilibrium, existence continues quietly, supported by adaptations that transform limited supply into sufficient sustenance. In this thin reservoir of breath, survival is not dramatic. It is steady, precise, and contingent upon conditions remaining within a narrow band. The telescopefish endures by matching itself to that band, living at the edge of what is available, yet never exceeding it, sustained by efficiency, shaped to meet the limits of its world. Change arrives in the ocean without sound. No wind announces it. No horizon marks its approach. It unfolds through gradual shifts in temperature, chemistry, and circulation, each adjustment subtle on its own, yet powerful in accumulation. Over time, these alterations reshape the layers that structure life below, modifying the conditions that define where creatures can exist. As global waters warm, the physical properties of seawater respond. Warmer water holds less dissolved gas than cooler water, including oxygen. Even small increases in temperature can reduce the amount available to organisms already living near their physiological limits. At the same time, warming strengthens stratification, the tendency of layers to remain separate rather than mixing. When mixing slows, oxygen from upper regions reaches deeper zones less efficiently, and renewal declines. These changes do not remain confined to chemistry. They influence movement throughout the water column. Many animals that travel vertically follow patterns tuned to long-established temperature gradients and oxygen distributions. When those gradients shift, the routes shift as well. Some species begin migrating at different depths. Others adjust the timing of their movements, and still others relocate entirely to remain within tolerable ranges. Such adjustments ripple outward, through ecological relationships. Predators track prey, prey track microscopic food sources. If one group alters its depth, the others may follow or fall out of alignment. The result can be a reorganization of community structure, not sudden but progressive, unfolding as generations respond to altered conditions. What once was a stable arrangement becomes fluid, its boundaries slowly redrawn. For highly specialized animals, flexibility may be limited. Species whose bodies and behaviors are precisely matched to a narrow set of environmental parameters cannot easily shift outside them. Temperature influences metabolic rate. Oxygen availability affects endurance. Light penetration determines how sensory systems perform. When these factors change together, the margin of suitable habitat may shrink. The telescopefish belongs to this category of precision. Its physiology and posture reflect fine-tuning to a particular combination of depth, illumination, and chemistry. Should warming waters alter those parameters, the zone where its adaptations function optimally could move or contract. It might descend farther, ascend slightly, or adjust activity patterns. Yet each option carries constraints set by anatomy and energy balance. Scientists studying ocean trends observe that some midwater habitats have already begun to shift. Instruments lowered through the sea, record temperature profiles, oxygen concentrations, and particle distributions over time. These measurements reveal gradual transformations in the vertical landscape, patterns that would be invisible without long observation. Data accumulates year after year, tracing how the interior ocean responds to atmospheric change. Within these shifting layers, life continues its quiet negotiations. Organisms encounter new boundaries where none existed before, or find familiar zones altered in subtle ways. Some adapt through behavioral plasticity. Others rely on genetic variation across generations. Still others may decline if conditions move beyond what their biology can accommodate. The fading of suitable conditions is rarely dramatic. It resembles twilight lengthening, a slow dimming rather than a sudden darkness. Yet the consequences can be profound. A reduction in oxygen may limit growth. A change in temperature may alter reproduction. A shift in depth may affect access to food. Each effect alone may seem modest, but together they shape survival. Thus, climate influence extends downward from surface air into remote water, linking distant processes through shared chemistry and circulation. The telescopefish drifts within that connection, its existence tied to balances that extend far beyond its immediate surroundings. As those balances evolve, the environment that shaped it continues to change, carrying with it the quiet challenge of persistence within a world that does not remain still. Far beyond coastlines and continents, an immense living system drifts in quiet suspension. It stretches across every ocean basin, surrounding the planet in a continuous band that few people ever witness. This realm is not defined by reefs or shorelines, but by depth, a vast midwater expanse filled with animals whose combined mass may rival, and in some estimates surpass, that of all fishes swimming in sunlit seas. Researchers who attempt to measure this abundance rely on indirect methods. Nets sample small portions. Acoustic instruments send pulses downward and interpret returning echoes. Submersible surveys record brief passages through limited volumes. Each approach offers fragments rather than totals, yet those fragments consistently suggest extraordinary density. The numbers hint at a population scale that challenges intuition, a hidden multitude inhabiting water once assumed sparse. Within this domain live countless species, many still unnamed. Some are delicate, some muscular, some nearly transparent, others armored with reflective scales. They occupy overlapping ranges, forming layered assemblages that shift with time and condition. The collective effect resembles a drifting continent of life, suspended between brighter waters above and darker depths below, extending across thousands of kilometers without interruption. Despite its magnitude, this ecosystem remains lightly studied. Compared with coastal environments or coral reefs, scientific attention here has been limited by distance, cost, and technical challenge. Expeditions require specialized vessels, advanced instruments, and long deployments. Even then, the area surveyed represents only a minute portion of the whole. Vast regions remain unsampled, their inhabitants known only through scattered traces. This limited knowledge leaves many questions unresolved, particularly regarding ecological roles and resilience. Scientists understand that these midwater communities participate in global cycles of nutrients and gases, yet the details of their interactions remain incomplete. Food webs interlace in ways still being mapped. Reproductive patterns, growth rates, and lifespans often remain estimates rather than measurements. The system's full complexity lies largely beyond direct observation. Interest from industry has begun to turn toward this unseen abundance. Advances in technology now make it technically feasible to harvest organisms from midwater layers that were once inaccessible. Some proposals focus on extracting small fishes and crustaceans for use in animal feed or nutritional products. The sheer scale of the biomass suggests potential yield, drawing attention from commercial ventures seeking new resources. Such interest introduces uncertainty. Removing large quantities of organisms from a system that has rarely been studied in detail carries risks difficult to predict. These animals form links within intricate networks, transferring energy between levels of the food web. If significant portions were removed, the effects could propagate through predator populations, nutrient cycles, and chemical balances. Yet, without comprehensive data, the magnitude of those effects remains unknown. Scientists and policymakers consider these possibilities with caution. International discussions explore how to evaluate impacts before large-scale extraction begins. Monitoring programs aim to establish baseline measurements against which future change could be compared. The goal is to understand this immense ecosystem before altering it, though the challenge is substantial given its size and complexity. Within this broad living field, the telescopefish drifts as one participant among multitudes. It is neither dominant nor insignificant, but part of a collective presence that shapes ocean function through sheer abundance. Its existence reflects the character of the midwater world itself, abundant yet concealed, influential yet seldom seen. To contemplate this hidden system is to recognize that much of Earth's life unfolds beyond ordinary awareness. The largest ecosystems are not always those most visible. Some lie suspended in remote layers, moving quietly, sustaining processes that reach from ocean depths to planetary balance. Not all signals arrive as light. In water, motion itself carries information, traveling outward as subtle disturbances that ripple through the surrounding fluid. Every fin stroke, every tail beat, every shift of a body sends faint waves across the medium. These waves move silently, yet persistently, spreading until they encounter another organism equipped to sense them. Along the sides of many fishes runs a sensory network known as the lateral line. It appears externally as a thin series of pores, but beneath those openings lies a chain of fluid-filled canals lined with microscopic receptors. Each receptor contains hair-like structures that bend when water moves across them. Even delicate displacement can trigger a response, translating motion into electrical signals that travel to the brain. Through this system, the telescope fish perceives events beyond direct sight. A nearby swimmer displaces water as it passes. A distant tail stroke produces a pressure fluctuation. These disturbances reach the lateral line and register as patterns of vibration. The fish does not see these signals, yet it senses their direction, strength, and rhythm, forming a spatial impression of activity around it. Pressure waves differ from light in one important respect. They do not require illumination. They move equally well through darkness, carrying information about objects regardless of visibility. For animals living in dim layers, this sensory channel provides a valuable supplement, extending awareness into regions where eyes alone might detect little. It allows perception of motion occurring outside the narrow visual field, offering early notice of approach or retreat. The receptors responsible for this detection are sensitive enough to distinguish fine gradations. Some respond to steady flow, others to sudden pulses. Together, they create a composite map of surrounding movement. Neural processing interprets these signals rapidly, enabling the fish to adjust posture or orientation without hesitation. The effect resembles a form of touch distributed across the body's surface, a tactile awareness mediated by water itself. Despite this additional sense, vision remains central. The lateral line indicates that something has moved, but it does not reveal shape or identity. It conveys direction and vibration rather than outline. To determine whether a passing disturbance represents prey, predator, or harmless drift, the fish still relies on its eyes. Visual information supplies detail, contrast, and confirmation that mechanical signals alone cannot provide. The two systems, therefore, operate together. Vibrational cues alert the fish to motion nearby. Visual input clarifies what that motion represents. One sense extends reach. The other refines interpretation. This partnership enhances survival without replacing the primary role of sight. The body integrates both streams seamlessly, combining them into a unified perception of its surroundings. Such redundancy offers resilience. If one channel becomes momentarily less effective, the other can still supply information. Turbulence may mask delicate pressure waves, while suspended particles may interfere with optical clarity. By maintaining multiple sensory pathways, the fish reduces reliance on any single source. Each sense supports the others, forming a network of perception distributed across different physical principles. Within this arrangement, the lateral line functions as an early messenger. It detects motion first, drawing attention toward a particular direction. The eyes then focus, examining the region indicated. Action follows only after both systems align, ensuring that response is guided by accurate interpretation rather than impulse. Thus, awareness in this environment arises from more than one sense working quietly together. Water itself becomes a carrier of information, and the body becomes an instrument tuned to receive it. Yet even with these additional signals, sight retains its guiding role, directing the final decision in moments that determine feeding or avoidance. The telescope fish moves through its world attended by vibration as well as light, supported by senses that complement rather than compete, each contributing to a perception shaped by the medium in which it lives. Life continues here, not through certainty, but through chance guided by rhythm. In a realm where individuals may drift great distances apart, meeting another of the same species is uncommon. The water is vast, and population density, though substantial on a planetary scale, is locally sparse. Each animal occupies its own quiet volume, carried gently by currents that seldom bring companions within immediate reach. Under such conditions, reproduction cannot rely on close courtship or prolonged pairing. Many midwater fishes appear to depend instead on broadcast spawning, a strategy in which eggs and sperm are released into the surrounding water. Fertilization occurs externally, the union of cells taking place within the drifting medium itself. This method allows reproduction without direct contact, increasing the probability that genetic material will meet even when adults remain separated. Timing becomes essential. For broadcast spawning to succeed, multiple individuals must release their gametes within a similar interval. If one releases too early or too late, the opportunity passes unused. The ocean offers no fixed calendar, yet subtle cues help synchronize these events. Changes in temperature, seasonal cycles of plankton abundance, or shifts in current patterns may act as signals. Internal biological clocks, shaped by generations of selection, likely respond to these environmental rhythms. The telescopefish is thought to follow such a pattern, though direct observation remains scarce. Scientists rarely witness spawning in this depth, and much of what is inferred comes from the study of related species. The fragility of deep-dwelling animals complicates experimentation, and their dispersal across immense spaces makes continuous monitoring difficult. Evidence accumulates slowly, gathered through examination of reproductive tissues or occasional sightings of eggs and larvae. Eggs released into the water drift freely, carried by currents that transport them away from their origin. This dispersal spreads offspring across wide areas, reducing competition among siblings and increasing the chance that some will encounter suitable conditions. The eggs are typically small and buoyant, containing just enough stored energy to sustain early development before feeding begins. Their transparency and delicate membranes help them remain inconspicuous within the surrounding fluid. Once hatched, larvae enter a phase of life distinct from adulthood, inhabiting different depths and feeding on different prey. During this stage, mortality can be high, as predators and environmental variability claim many individuals before maturity. Yet the strategy does not depend on each offspring surviving. Instead, it relies on numbers, releasing many eggs so that a fraction may persist and continue the lineage. The absence of direct encounters between adults means that reproduction unfolds as a quiet exchange dispersed through space. No elaborate display marks the moment. No structure shelters the young. The ocean itself becomes the nursery, its currents distributing developing forms through shifting layers. Success is measured not by proximity, but by synchronization, the alignment of biological timing with environmental opportunity. Researchers continue to investigate these processes, seeking clearer understanding of how often spawning occurs, how many eggs are released, and what conditions favor survival. New instruments capable of sampling genetic traces from seawater offer promising clues, revealing the presence of species even when individuals are not seen. Such methods may gradually illuminate patterns that have long remained hidden. Within this system, continuation depends on delicate coordination between organism and environment. The telescopefish contributes to that continuity through a reproductive strategy suited to distance and darkness, releasing the possibility of future generations into the surrounding water. In the vastness that separates individuals, life persists through timing, dispersal, and the quiet meeting of cells carried together by the sea. Time moves differently where light is scarce and food uncertain. Without strong seasonal contrasts or rapid shifts in temperature, the deep ocean unfolds in steady gradients rather than sharp cycles. Within this measured environment, many midwater species grow slowly, their bodies adding mass in increments so gradual that change is nearly imperceptible from one year to the next. The telescopefish follows this restrained tempo. Growth is not driven by abundance but moderated by limitation. Each meal contributes only modestly to overall size, and long intervals between feedings slow the accumulation of tissue. Metabolism remains subdued, allocating energy first to maintenance of essential systems before investing in expansion. As a result, maturation may require years rather than months, with developmental stages extending across extended spans. Estimating lifespan in such species presents difficulty. Direct observation over decades is rare, and tagging individuals at depth remains technically challenging. Scientists often infer age through indirect methods, examining growth rings in otoliths, the small calcified structures within the inner ear that record incremental deposition over time. In some deep-dwelling fishes, these rings suggest lifespans considerably longer than those of comparable surface species. Though specific data for the telescopefish remain limited, its physiology aligns with patterns associated with longevity. Slow metabolism tends to correlate with extended lifespan, as cellular processes proceed with reduced oxidative stress and lower cumulative wear. Energy is conserved carefully, and repair mechanisms operate without the rapid turnover seen in fast-growing animals. This quiet pace may allow individuals to persist for decades within stable conditions. Living across such extended periods requires disciplined energy budgeting. Every movement, every reproductive event, every metabolic reaction draws from finite reserves gathered through uncertain feeding. The fish must balance immediate needs against long-term survival, ensuring that present expenditure does not compromise future resilience. This balance is maintained through hormonal regulation and metabolic pathways tuned for efficiency rather than speed. Reproductive timing reflects this extended horizon. Instead of investing heavily in a single season, the fish may reproduce multiple times over its lifespan, distributing effort across years. Such a strategy reduces risk, allowing success in one period to compensate for failure in another. Longevity thus becomes an asset, increasing the probability that favorable conditions will eventually coincide with reproductive readiness. The surrounding environment supports this slow existence. Temperatures remain relatively constant at depth, reducing physiological stress associated with fluctuation. Predation pressure, while present, differs from that in shallower waters where visibility and competition are greater. The overall rhythm favors endurance, rewarding individuals capable of sustained efficiency over rapid turnover. Yet extended lifespan also binds the species closely to environmental stability. Animals that mature slowly and reproduce across decades may struggle to adapt quickly if conditions shift abruptly. Their long generational intervals mean that evolutionary change proceeds at a measured pace. Resilience depends not only on individual endurance, but on the constancy of the habitat that shaped them. Within this quiet chronology, the telescopefish inhabits a world where days accumulate into years without dramatic markers. Its life unfolds in slow increments, measured by growth that scarcely announces itself and by feeding events spaced across extended intervals. Time is neither hurried nor wasted. It is absorbed steadily, converted into maintenance, survival, and eventual reproduction. In such a setting, longevity is not an accident but an outcome of alignment between physiology and environment. The fish endures by matching its internal clock to the unhurried cadence of the deep, living not in bursts of rapid change but in sustained continuity, its existence stretching across years shaped by patience and careful use of energy. Perception is shaped long before conscious thought begins. The human mind interprets unfamiliar forms by comparing them with patterns it already knows, drawing on a lifetime of faces, animals, and movements that follow familiar arrangements. When something appears outside those expectations, the brain pauses, searching for a category that fits. If no clear match is found, the impression can feel strange, not because the object is unnatural, but because it does not align with stored templates. Most vertebrates encountered in daily life share a common visual arrangement. Eyes face forward or slightly to the sides, positioned within proportions that balance symmetry and expression. Humans, primates, birds, and many terrestrial animals follow variations of this design. Through repeated exposure, the brain learns to treat such layouts as normal. Neural circuits specialized for facial recognition reinforce this familiarity, responding most easily to patterns that resemble those already memorized. When confronted with a form organized differently, those circuits must work harder. The telescopefish presents such a case. Its sensory structures occupy positions that depart from expectations shaped by land animals and surface dwellers. The resulting impression can seem unsettling, not because the arrangement lacks purpose, but because it challenges the perceptual shortcuts the brain prefers to use. Psychologists describe this response as a consequence of predictive processing. The mind continuously anticipates what it is about to see, using prior experience to generate internal models. Incoming sensory information is compared with those predictions. When the match is close, recognition feels effortless. When the match diverges, attention sharpens as the brain updates its model. The sensation of unfamiliarity arises during that adjustment. This reaction reflects human bias rather than biological flaw. We tend to evaluate other organisms according to standards derived from our own anatomy and environment. Features that resemble our expectations appear orderly, while those that differ seem unusual. Yet such judgments reveal more about the observer than about the observed. Nature does not share our preferences for symmetry or proportion. It favors whatever configuration allows survival within a given set of conditions. The idea that evolution seeks beauty is a projection. Natural selection operates through differential success in reproduction and persistence, not through aesthetic criteria. Structures persist because they function effectively, not because they conform to human notions of elegance. In many cases, the most efficient design within a particular habitat may appear awkward or exaggerated when viewed outside that context. Our sense of the uncanny often arises when something is almost familiar yet not quite aligned with expectation. A slight deviation in proportion or placement can draw attention more strongly than a completely foreign shape. The telescopefish occupies that threshold. It is recognizably a fish, yet its configuration diverges just enough from common forms to disrupt automatic recognition. The brain lingers on the discrepancy, attempting to reconcile it. Over time, repeated exposure can soften this reaction. As new patterns become familiar, neural pathways adjust, and what once seemed strange begins to appear coherent. Understanding also plays a role. When the observer learns how each feature contributes to survival, the form acquires context, and context replaces surprise with comprehension. The unfamiliar becomes intelligible. In this way, perception evolves within the observer, just as anatomy evolves within the organism. Both processes involve adaptation, though on different scales of time. The telescopefish remains unchanged in its design, yet the human response to it can shift as knowledge deepens. What once seemed improbable resolves into logic shaped by environment. Seen through this lens, the feeling that such a creature should not exist reveals the limits of expectation rather than any flaw in nature. The fish persists because it suits its world precisely. Our surprise simply marks the moment when human assumptions encounter a design formed under conditions far removed from our own. Adaptation can narrow as much as it empowers. When a lineage refines itself toward a single solution, it gains efficiency within that solution's boundaries. Every structure aligns with a particular demand, every behavior tuned to a specific circumstance. The result is elegance of function, yet also confinement, because excellence in one direction often limits flexibility in others. The telescopefish embodies such refinement. Its visual system, posture, and metabolism converge upon a precise environmental window. Within that window, it performs with quiet effectiveness. Its eyes gather faint contrast with remarkable sensitivity. Its body conserves energy across long intervals. Its feeding strategy depends on timing rather than pursuit. These traits combine into a cohesive whole, a design honed for the twilight layer it inhabits. This degree of specialization grants strength. By committing fully to a narrow niche, the fish avoids competition with species adapted to broader ranges. It occupies a position shaped for its unique combination of depth, light, and prey movement. Efficiency reduces waste. Alignment reduces uncertainty. Within stable conditions, such precision can persist across generations with little need for modification. Yet, specialization carries inherent risk. A narrow niche depends on the continued existence of that niche. If light levels shift, if oxygen boundaries move, if prey distributions alter, the finely balanced system may find itself outside optimal range. A generalist species can sometimes adjust behavior or habitat more readily, drawing upon wider tolerances. A specialist must either adapt through evolutionary change or decline. Resilience, in such cases, depends on the breadth of variation within the population. Even highly specialized species retain some genetic diversity, small differences among individuals that may become significant under new conditions. If environmental pressures change gradually, selection can favor variants better suited to emerging realities. Over time, the population may shift, preserving function while altering detail. Fragility arises when change occurs faster than adaptation can proceed. Long generation times and slow growth may limit the pace at which evolutionary adjustment unfolds. The very traits that enable endurance within a stable system, such as measured metabolism and extended lifespan, can slow responsiveness to rapid disruption. The balance between strength and vulnerability thus hinges on tempo, the rate at which environment transforms relative to the rate at which life can respond. This tension is not unique to one species. Across ecosystems, organisms occupy positions along a continuum between flexibility and specialization. Coral reefs, polar ice communities, desert flora, each demonstrate variations of the same principle. Success within a defined set of conditions may entail exposure when those conditions shift. Nature does not favor one strategy universally. It favors what works in context. For the telescopefish, context has long remained consistent enough to sustain its design. The twilight zone has persisted over geological timescales, offering continuity upon which specialization could build. Within that continuity, precision became advantage rather than liability. The fish's form represents a long negotiation between opportunity and constraint, resolved through refinement. Considering this balance invites a broader understanding of adaptation itself. Specialization is not excess but commitment, an investment of biological resources into a particular solution. It can produce extraordinary efficiency, yet it asks for environmental stability in return. When that stability holds, the specialized thrive. When it wavers, the limits of their design become apparent. In the quiet depths where this fish drifts, strength and risk remain intertwined. Its narrow focus grants mastery within its chosen corridor. Its narrow focus also binds it to that corridor's persistence. The interplay of resilience and fragility continues, written into anatomy and circumstance alike, shaping a life that endures through precision balanced against uncertainty. Knowledge thins in the midwater, much like the light that filters down from above. For all that has been gathered, cataloged, and measured, vast portions of this ecosystem remain described in outlines rather than detail. The telescopefish, despite its distinctive form, exists within that partial understanding, its life traced through fragments of observation and careful inference. No reliable census defines its population size. The twilight zone stretches across the global ocean, immense in volume and uneven in structure. Sampling such an expanse presents formidable challenges. Nets collect only a fraction of what drifts within their path, and avoidance behaviors may allow agile individuals to escape capture. Remote vehicles illuminate small windows of space, revealing brief glimpses rather than comprehensive counts. From these scattered encounters, abundance must be estimated indirectly, often through extrapolation rather than direct measurement. Behavior, too, is largely reconstructed from anatomy and context. The upward orientation of its eyes suggests a hunting strategy shaped by vertical contrast, yet continuous observation of feeding sequences remains rare. The fish's posture, muscle structure, and jaw alignment imply a measured approach rather than rapid pursuit, but most accounts rely on preserved specimens or fleeting footage. Without sustained monitoring in its natural setting, much of its daily routine remains inferred. Feeding frequency illustrates this uncertainty. In environments where energy arrives unpredictably, consumption may be episodic rather than regular. Stomach content analysis offers snapshots, revealing what was eaten shortly before capture, yet such samples cannot reveal the intervals between meals. Metabolic models provide estimates based on body size and temperature, indicating how much energy must be acquired over time, but the rhythm of acquisition, whether steady or sporadic, remains unclear. Genetic studies, which might clarify population structure and connectivity, are limited by access to high-quality tissue samples. Deep-sea organisms often suffer damage during retrieval, complicating molecular analysis. Even when samples are viable, assembling comprehensive datasets requires coordinated international effort and consistent funding. As a result, the genetic diversity within and between regions remains only partially mapped, leaving open questions about how isolated or interconnected populations may be. Reproductive patterns are similarly indistinct. While broadcast spawning is considered likely, direct confirmation is scarce. The timing of gamete release, the survival rates of larvae, and the routes by which juveniles descend to deeper layers are topics of ongoing speculation. Without longitudinal studies tracking individuals across life stages, these processes remain outlined but not fully described. Technological advances promise gradual refinement of understanding. Autonomous vehicles capable of extended deployment can survey larger volumes of water with minimal disturbance. Environmental DNA sampling may detect presence through trace genetic material suspended in seawater, offering clues to distribution without requiring capture. Acoustic methods can sometimes register movements of midwater communities, hinting at biomass patterns otherwise hidden from view. Each innovation expands the window slightly, yet the view remains incomplete. Uncertainty does not diminish significance. Instead, it underscores the scale of the system in which the telescopefish participates. The midwater realm operates largely beyond direct human perception, and scientific exploration proceeds step by careful step. Every confirmed detail rests upon years of incremental work, and each new dataset refines previous assumptions. In this ongoing process, absence of certainty becomes part of the narrative. The fish continues its life, whether observed or not, its patterns unfolding within conditions that shaped them long before instruments descended. What remains unknown invites continued attention, not as a gap to be hurriedly filled, but as recognition that even in an age of satellites and submersibles, large portions of the ocean still move beyond complete comprehension. Direction shapes perception as surely as anatomy does. When a sensory system favors one orientation, it inevitably neglects another, not through failure but through choice embedded in structure. The telescopefish lives with such a choice inscribed into its design, its entire visual arrangement aligned toward a single axis that defines how it encounters the world. Its sight is concentrated rather than expansive. Instead of surveying a broad panorama, it perceives a restricted corridor that extends upward through the water column. Within that corridor, sensitivity is exceptional, capable of detecting slight contrasts and faint movements. Outside it, awareness diminishes. Objects drifting beyond this visual channel may pass unnoticed unless they create disturbances detected through other senses. The arrangement resembles a spotlight fixed in place, bright along its beam, yet leaving surrounding space comparatively dim. This narrowing represents a trade shaped by evolutionary pressure. In habitats where certain directions contain more useful information than others, selection can favor sensory investment toward those directions. Resources devoted to enlarging the upward field might reduce those available for lateral vision. Over generations, incremental changes accumulate, gradually emphasizing one region of perception while allowing others to recede. Such specialization confers clear advantages when environmental signals are predictably oriented. The telescopefish can devote neural processing power to interpreting stimuli from above, refining detection where it matters most. Concentration increases clarity. Signals entering that region receive immediate attention, their details resolved with precision. The cost lies in reduced awareness elsewhere, a limitation accepted in exchange for heightened performance within the chosen range. Reduced side awareness alters behavior as well as perception. Movement must remain measured, minimizing exposure to directions not fully monitored. The fish's posture supports this caution, maintaining alignment with its primary viewing axis. Even subtle adjustments of position can shift what enters or leaves its perceptual corridor, so orientation becomes integral to vigilance. The body itself participates in seeing, holding steady to preserve the alignment that grants effective vision. Evolution rarely eliminates such compromises. Instead, it balances them, weighing benefit against constraint. A wider field of view might allow detection of more surroundings, yet it could also dilute sensitivity where precision is essential. A narrower focus heightens detail, but restricts coverage. Natural selection does not pursue perfection in every dimension. It settles for configurations that succeed overall within specific conditions, even if they impose limits in others. Other sensory systems help compensate, providing information from directions outside the visual channel. Vibrational cues may reveal approaching motion. Chemical signals may hint at nearby organisms, and subtle pressure changes may indicate displacement in the water. These inputs supplement vision, though they do not replicate its detail. They form a secondary network that fills gaps left by optical specialization, reducing vulnerability while preserving the benefits of focused sight. Still, the compromise remains fundamental. The telescope fish perceives its surroundings through a lens shaped by necessity, not completeness. What it gains in acuity, it relinquishes in breadth. This balance reflects the principle that every adaptation carries both power and limitation, each inseparable from the other. In its quiet domain, the fish persists within that balance. It neither possesses nor requires total awareness. Instead, it holds mastery over the direction most relevant to its survival, allowing less critical regions to fade into lesser attention. The cost of looking upward is therefore not loss, but exchange, a deliberate allocation of biological resources toward a single visual pathway that defines how this creature engages with its environment. At first encounter, the form appears improbable, as if assembled from mismatched ideas rather than shaped through gradual refinement. The proportions do not align with familiar expectations, and the arrangement of features resists quick interpretation. Yet unfamiliarity can conceal precision, and what seems unlikely at a glance may reveal coherence when viewed through the conditions that produced it. The telescope fish exists as an answer rather than an anomaly. Its structure reflects a particular problem posed by its surroundings, a problem defined not by predators or currents alone, but by perception itself. In an environment where usable visual information arrives from a limited direction, survival depends on detecting that information with maximum clarity. The fish's anatomy represents a response shaped through countless generations, each adjustment favoring individuals whose sensory alignment match the demands of their habitat. This solution is narrow but effective. Instead of maintaining a generalized design capable of functioning moderately well in many circumstances, the fish embodies a configuration tuned for one circumstance in particular. Its orientation, physiology, and behavior converge upon a single objective, extracting meaningful signals from a setting where such signals are scarce. Every feature contributes to that purpose, forming an integrated system that transforms a difficult condition into a workable one. To observers accustomed to different environments, the result may seem unusual. Human perception tends to equate familiarity with normality, and deviations from common patterns can appear improbable. Yet probability in nature is measured not by resemblance to expectation, but by compatibility with context. A structure that looks unlikely in one setting may be entirely appropriate in another. What appears strange is often simply specialized. The fish's persistence demonstrates that its design meets the requirements imposed upon it. It continues to feed, grow, and reproduce within a region where many organisms would struggle to function. Its success does not depend on speed, ornament, or breadth of capability. It depends on alignment between form and circumstance, a correspondence refined until excess features fell away and essential ones remained. Such refinement illustrates a broader principle of evolution. Natural selection does not aim for versatility unless versatility improves survival. When a single challenge dominates, solutions may become increasingly focused. Over time, that focus can yield organisms whose bodies resemble instruments calibrated for a specific measurement, sensitive within their range, yet unsuited to others. This outcome is neither accidental nor extravagant. It is the predictable result of sustained selection acting upon variation. The telescope fish therefore stands as a testament to the logic of adaptation. Its appearance does not signal impossibility. It signals resolution, the settling of biological possibilities into a configuration that addresses a distinct need. What might seem improbable from an external perspective becomes inevitable when traced through the sequence of pressures that shaped it. Understanding this reframes the initial impression. The fish is not a curiosity placed outside ordinary patterns. It is a participant in those patterns, shaped by the same processes that formed countless other species, differing only in the particular demands it encountered. Its design follows rules that apply across life, even if the outcome appears unfamiliar to human eyes. Within its own environment, no contradiction exists. The features align, the system functions, the animal endures. What seems unusual from afar resolves into practicality when considered within context. The telescopefish survives not despite its distinctive form, but because of it, its body embodying a precise answer to a challenge presented by its world, an answer written in structure rather than explanation. Maps often suggest completion. Lines trace coastlines, colors mark depths, and names label regions as though the ocean were fully known. Yet, these charts conceal how little direct exploration has reached the interior waters that fill most of the planet. Vast stretches remain visited only briefly or not at all, their inhabitants recorded through scattered encounters rather than continuous study. Estimates indicate that a small fraction of the deep ocean has been observed closely by human instruments. The percentage varies with method and definition, yet all assessments agree on one point. Most of this realm lies beyond thorough examination. Remote cameras descend for hours. Autonomous probes drift along programmed paths. Research vessels survey selected coordinates. Even with these efforts, the explored portion remains tiny compared with the total volume. The twilight zone stands at the threshold of this unknown. It is neither near enough to the surface for easy access, nor deep enough to attract the same attention given to the ocean floor. Its depth complicates routine observation, and its breadth resists quick surveys. As a result, it persists as a frontier within reach, yet still largely uncharted, a region where discovery continues to outpace documentation. Creatures inhabiting this band exemplify that frontier. Many species are known from only a handful of specimens. Some are described from single captures, their biology inferred from anatomy rather than witnessed directly. Others appear in camera footage without ever being collected, leaving scientists to classify them provisionally while awaiting clearer evidence. Each new expedition adds pieces to a mosaic that remains incomplete. The telescopefish represents this condition of partial knowledge. Its existence is confirmed, its general features understood, yet much about its daily life remains hidden. It serves as a reminder that even distinctive organisms can remain elusive when they occupy environments difficult to access. Its silhouette has entered scientific records, yet the details of its habits continue to unfold gradually through patient investigation. Frontiers in science differ from those of geography. They are not marked by borders, but by uncertainty, by regions where observation has not yet resolved questions into answers. The twilight zone embodies such a frontier, not distant in miles, but distant in understanding. Instruments must withstand pressure, darkness, and isolation. Data must be interpreted cautiously, aware that each measurement samples only a small portion of a vast system. Exploration here proceeds through accumulation. A single expedition yields observations. Multiple expeditions reveal patterns. Over years and decades, fragments combine into clearer pictures. Progress may seem slow, yet it reflects the scale of what is being studied. The ocean interior is immense, and its processes unfold over distances and times that challenge human schedules. Symbols often arise to represent unknown realms. In earlier centuries, distant mountains or polar ice carried that role. Today, midwater ecosystems hold a similar place in scientific imagination. The telescopefish, with its unusual form and hidden life, stands as one such symbol, not because it is rare alone, but because it illustrates how much remains unseen even among creatures already discovered. To consider this is to recognize that knowledge does not end at the boundary of current understanding. Beyond that boundary lies a field of possibilities awaiting careful observation. Each descent of an instrument, each recovered sample, each recorded image extends that boundary slightly farther. The frontier shifts not by conquest, but by attention. Within the dim layers where this fish moves, the unknown persists alongside the known. Facts exist beside mysteries. Certainty coexists with questions yet unformed. The twilight zone continues to hold its distance, inviting inquiry without yielding all at once, a quiet expanse where discovery advances in measured steps and where the edge of human knowledge remains gently in motion. In the deep ocean, design follows necessity with unhurried precision. Forms arise not from spectacle but from constraint, each contour guided by forces that persist year after year. Pressure shapes tissues into flexible resilience. Scarce light directs sensory investment toward faint signals. Limited energy encourages restraint in motion and growth. Within this convergence of factors, structure becomes a record of influence, written slowly into bone and muscle. The telescopefish stands within that record. Its body reflects negotiations carried out across generations, adjustments so gradual that no single moment marked their arrival. A slightly altered eye angle offered clearer detection. A marginal shift in jaw placement improved capture. A more efficient metabolism extended survival between meals. None of these changes were dramatic in isolation, yet together they formed coherence, an arrangement that fits its surroundings with quiet exactness. Nothing about such a creature is random. Variation arises continuously in populations, but only certain variations persist. Those aligned with environmental realities endure. Others fade. Over time, the remaining features appear purposeful because they are filtered through consistent conditions. What remains is what works. In this way, inevitability emerges not from intention, but from repeated selection acting within stable boundaries. To observers from above, the result can seem improbable, as though such a form could not reasonably exist. Yet improbability depends on perspective. Within the context of its habitat, the telescopefish represents a logical solution to overlapping pressures. Darkness narrows visual opportunity. Vertical movement of prey creates directional patterns. Energy scarcity rewards efficiency. The fish embodies the intersection of these demands, neither excessive nor deficient, but proportioned to match them. The deep ocean reinforces this principle across countless species. Organisms there reflect combinations of temperature, chemistry, movement, and time that differ from those near the surface. Adaptations accumulate accordingly. Some species emphasize transparency, others develop bioluminescent signaling, still others refine sensory structures toward specialized tasks. Each design corresponds to a particular arrangement of constraints, forming a mosaic of solutions rather than a uniform template. Human experience occupies a thin layer of this mosaic. Most daily encounters occur in environments rich with light and oxygen, where movement is unconstrained and resources comparatively abundant. From that vantage, the deep can appear distant, even alien, yet it constitutes a vast proportion of the planet's living space. Beneath familiar horizons lies a domain where biological rules continue without regard for surface expectations. In that broader view, the telescopefish becomes less an exception and more an example. It illustrates how life adapts when confronted with persistent limits. Its features are not extravagant deviations, but measured responses, each aligned with environmental constants that have endured for millennia. The logic behind them is steady, cumulative, and indifferent to aesthetic judgment. Most of Earth's biosphere unfolds beyond ordinary sight. Midwater ecosystems, abyssal plains, and subsurface habitats support organisms rarely encountered directly. Their existence does not depend on visibility. They function within systems that circulate nutrients, regulate climate, and sustain complex food webs. The surface represents only a fraction of this living network, a bright veneer atop deeper continuities. Recognizing this scale shifts perception gently. The unusual becomes contextual. The improbable becomes consistent with pattern. The telescopefish persists not as a challenge to understanding, but as confirmation of it, demonstrating that when pressure, light, and hunger converge in a particular configuration, life answers with corresponding form. In the quiet logic of the deep, such outcomes are not surprising. They are the natural result of time, constraint, and the persistent capacity of living systems to align themselves with the worlds they inhabit.